Welcome back to the Bioinformatics Practical series, and today we talk about how to find potential protein coding regions in a sequence, right? And the simplest method of doing that is by finding what is known as the open reading frames. So before moving on to what is an open reading frame, let me give you a background of genome annotation. So let's start with genome annotation first, and then of course we discuss the simplest method of finding potential protein coding regions using the open reading frame finder. And so our story would begin with the discovery of the genetic code. And the first important thing that I want to give you as a concept is the features of the genetic code. So the important features of genetic code include universal, that it is a common genetic code across all. Of course, there are minor variations that may happen, but generally the genetic code is universal. Second most important point here is that the codon is a triplet, or three nucleotides together code for a single amino acid, right? And you know that since there are 4 nucleotides available and there are 3 positions where you can have a nucleotide of each type, the total number of codons available is 64. So you have 64 codons lined up here, of which one is the common start codon, which is AUG. In some cases, GUG may also act as a start codon, but the standard start codon is AUG, which codes for methionine. And then we have 3 stop codons, include your UAA, UAG, and UGA, right? Apart from these, there are other 61 codons, including AUG, that can potentially code for a amino acid. And again, what is important is since there are 61 codons and 20 amino acids, many amino acids can have more than one codon to code for them, right? The other important point is the code is unambiguous. A given codon will always code for the same amino acid. One codon, one amino acid correspondence is absolute. Another important point here is you can see that there are more than one codons coding for the same amino acid. For example, your leucine is coded by UUA, UUG, CUU, CUC, CUA, and CUG. So this essentially is that while one codon always codes for the same amino acid, one amino acid can be coded by multiple codons. So this is what is known as the degeneracy of the code. So 4 important features of the genetic code include universality, and the codon is a triplet. The code is unambiguous, means one codon codes always for the same amino acid, and the code is degenerate, meaning that one amino acid may be coded by several different codons. So now let's also have a look at the structure of a simple prokaryotic gene sequence. So here you are. This is your typical gene sequence for a prokaryote. You'll have a transcription start site where the RNA polymerase will come and bind and start the transcription. You'll have a 5' UTR or untranslated region, followed by the start codon where the actual translation begins. So this is your translation start site, and then followed by several codons in order until it comes to a point where there's a stop codon, which indicates that the translation has to stop here. And beyond that, you have the 3' UTR on the 3' untranslated region. -10 and -35, you have promoter sequences, which are simple proximal control elements in prokaryotes. There is no split gene structure, which means the exon-intron structure is not found in prokaryotes. Other important point is that genes of the same pathway in prokaryotes are organized together into what is known as operons. So these are structurally and functionally clustered regions of the gene. And then, of course, you have the regulatory regions are relatively simple and more conserved in prokaryotes. So therefore, the genome annotation or identifying protein coding regions in a prokaryotic genome is a lot easier as compared to a eukaryotic genome. So let's now talk of a simple sequence and try and find out the genes present in that sequence. This is your cloning vector PBI322 complete sequence taken down from NCBI, and this is the FASTA format. You can see you have a greater than sign followed by accession number and a small detail. This is cloning vector PBI322 complete sequence, and this is followed by the sequence itself straight away, right? Now, if you look at the sequence here, sequence alone may not mean much because you don't know exactly which nucleotide is the start of a gene, which nucleotide is the end of gene. So here I show you a DNA sequence without any annotation, which means you do not know where the genes are. So raw sequence data by itself does not mean anything unless it is annotated. What do you mean by annotation? Annotation means adding structural and functional features to regions of the genome. So for example, if you take the example of human genome, one common annotation would be the size of the human genome, 3.2 billion base pairs. So this is one annotation. The number of genes in the human genome, around 24,000, a second annotation. A given gene, how many isoforms, and whether it undergoes alternative splicing or not, another level of annotation, right? The expression pattern of the gene and its isoforms in different tissues is another next level of annotation. What is the methylation state or the epigenetic state of the gene? Another track of annotation. So annotation is endless, and as and when we gain information about a gene, and as and when we gain information about biological processes, we tend to add annotation layers, or the layers of annotation. For example, in this sequence, you have these green lines indicate genes and the arrows indicate their orientation. So this is the 5' end, this is the 3' end, and some genes are on the top strand, other genes on the bottom strand, and so on and so forth. So our job using ORF Finder is to find out potential protein coding regions in the given DNA sequence. Before we move on to finding the potential protein coding regions, there is a concept called reading frames in a DNA. So let's have a look at the concept of reading frames first, and then we move on to open reading frames. So here you are, if you look at this DNA sequence here, this is the 5' end, this is the 3' end. You know that codon is a triplet, so basically this DNA sequence has to be read in blocks of 3 each, right? So if you start counting from the first nucleotide here, you can have blocks of 3, for example, AGG, TGA, CAC, CGC, AAG, CCT, TAT, ATT, AGC, and so on and so forth if the sequence continues. So this is what is known as a reading frame 1, because you're starting with the first nucleotide and taking blocks of 3 together. Then you could have, likewise, you could have a reading frame 2, where you start reading from the second nucleotide and then again take blocks of 3. So now this becomes GGT, GAC, ACC, GCA, AGC, CTT, ATA, TTA, GC, and so on and so forth. Likewise, you could start with the third nucleotide and again take blocks of 3 and get your third reading frame, which is this one here. Now, if you see, depending on which reading frame you're looking at, the codons have absolutely changed, and because they have absolutely changed, the protein that they're going to code are also going to change. So now if you start reading from the fourth nucleotide onwards, so this was TGA, CAC, CGC. So basically it is the reading frame is still 1 minus the first codon. So this is TGA, CAC, CGC. You can enjoy the bird sound for a moment. You also know that the DNA is double-stranded, and you could have reading frames on both the strands. So therefore, for a given DNA sequence, there are 6 reading frames possible. So any fragment of DNA has 6 reading frames and can potentially code for 6 different polypeptide sequences. So now we pick up the concept of open reading frames. So here is a sequence again, which is given to you: 5' ATG, AA, CTG, TAT, GCT, CAT, TTA, ACT, and TAG, right? And this is the 3' end here. And we know that our start codon is AUG, and our stop codons include UAA, UAG, and UGA. What we are interested in is in a reading frame that gives us a start codon, followed by several codons, followed by a stop codon. If you look here in this sequence, you have a start codon here, and you have a stop codon here, and in between you have several different codons. So this actually is itself a complete sequence itself as an open ending frame minus a stop codon. Now, if I were to ask you, can you see another open ending frame within this sequence? So if you look closely here, this is an open ending frame again. You have a start codon here. You have ATG followed by CTC followed by ATT and followed by TAA, which is a stop codon, right? So this is another open ending frame within this region here. Of course, it is not in the first reading frame. It would be in the second reading frame, if you see, right? So the top open ending frame is actually coming from the first reading frame. So because you start from the first nucleotide in the sequence, and the second open ending frame is coming from the second reading frame, if you see, right? So one of the simplest methods of finding potential protein coding regions is to find an open ending frame, or ORF. A sequence of nucleotides is starting with the start codon, followed by a threshold number of codons in between, and ending in stop codon is what is known as the open reading frame. Importantly, the stop codon is not a part of the ORF, right? So here in this case, you have two open reading frames. One is in the reading frame 1, and the entire sequence itself is an open reading frame. The other is in reading frame 2, and it is basically a smaller reading frame here. So what is also important here is to mention that you need to put up the threshold number of codons that must be there before you can define that this is an open reading frame. If you put the length as too short, you may have problems in the sense that you may find too many regions in the genome that may satisfy the criteria of open reading frame, but they may be too short and actually not coding for the gene. Another important point here in this given sequence is that there are two open reading frames. One is slightly larger and the other is slightly smaller. So if you were to predict a potential protein coding region in this region, it is most likely that the longest open reading frame is the correct reading frame, because it has been so conserved. Unless it had a function, it would not have been conserved this way. So the idea is, if within a given region you have two different open reading frames possible in different frames, then the longest one is most likely to be correct. So let's now look at the properties of open reading frames. So I've already explained that the length of the ORF is critical to annotate a gene, right? Long open reading frames are more likely to be genes. So for a given region, if there are two different open reading frames possible in different reading frames, then the longest ORF is likely to be the most correct, and because it has been conserved as such and therefore implies functionality. So a basic approach to identify potential protein coding regions in a sequence is to scan the genome for open reading frames with a certain threshold length. So this length cannot be too short, else you'll have a lot of false positives. So this length setting is critical, because if it is too short, you'll have a lot of false positives. If it is too high, you may have a lot of false negatives. So both have to be avoided. And in prokaryotes, greater than 90% of the genome codes for proteins, and therefore prediction of ORFs greater than 100 bases is a fairly good indication of putative gene. And in case of multiple ORFs in the same region in different frames, the longest one is most likely to be the correct one. And it will also be in context to tell you the approaches to genome annotation. And first, of course, is the statistical or ab initio method, or the de novo method, where you're not comparing it to any known sequence. You're directly studying the statistical properties of the sequence to identify potential protein coding regions. ORF Finder would also come in this category of statistical or ab initio methods. Then there is a second method of identifying potential protein coding regions in a given DNA sequence. This is why comparison to another similar DNA sequence which has already been annotated. So largely this is done on the basis of sequence similarity, and sequence similarity could be to DNA, could be to mRNA, or it could be to protein, right? And the third idea is what is known as the combined evidence approach, where you use first the statistical or ab initio method to identify potential protein coding regions, and then you do a homology-based inference to see whether the regions that are predicted by your program as potential protein coding regions are actually true or not. So basically what we're trying to see is that if giving a sequence, I predict an open reading frame, and comparing it to known protein sequences to see if there is a similar sequence which is already annotated. So this will help me functionally annotate my sequence. While I know that this is my start and end of the gene, I still do not know what is the function. So if there is another similar protein which is pre-annotated and whose function is known, I can directly take that function and put it up here as a function of my predicted open reading frame, right? So this is what is known as combined evidence approach, and the current method of open reading frame finder in NCBI allows you to first do an in silico or ab initio prediction of open reading frames, and then it also allows you to do a homology-based confirmation of the sequences that are predicted as open reading frames of potential protein coding regions. So the ORF Finder combines both statistical or ab initio method and the homology-based methods, and also allows you to confirm whether a prediction is true or not. So this is your ORF Finder, ncbi.nlm.nih.gov/ORF Finder/, that's the URL, and ORF Finder searches for open reading frames in the DNA sequence you enter. The program returns the range of each ORF or open reading frame along with its protein translation. use ORF Finder to search newly sequenced DNA for potential protein coding segments. And you can verify whether these protein coding segments are actually true or not by using the newly developed Smart Blast or regular Blast P, which will match your predicted ORF with the known protein databases, right? This web version also, as happens with most of the web versions, there is a limitation to the length of the sequence that can be annotated here. But should you have a standalone or command line version, you will not have a problem. And only thing is, it must also have a Linux 64 operating system. Now we'll paste the sequence here. So let me first pull out the sequence for you. And what we'll do is we'll take the sequence of PBR322 and see whether the ORF Finder is able to find the two main genes in PBR322. That is the tetracycline resistance and ampicillin resistance genes. So what we're going to do here is to paste the sequence of PBR322, and we know in PBR322 there are two main resistance genes, the ampicillin resistance gene and the tetracycline resistance gene. So we would like to check using ORF Finder whether the ORF Finder is able to find these genes in the plasmid sequence that we're inputting here, right? So let me first pull out the sequence for PBR322. So we go to NCBI again. In the all databases, I'll select for nucleotide sequence. Here you are, and I give it the search term P capital BR 322. Right. So here you are, and this is your PBR 322 complete sequence. Let me click this and get the sequence here, right? So you could directly get it. We by default will get the GenBank format, and in the GenBank format, you know there will be annotation and also the sequence downstream. So if you look into the annotation, the tetracycline resistance gene is found from nucleotide 86 to 1276. And the product is tetracycline resistance protein, right? And this is the protein translation for that, if you see. And then if you go down further, you'll find the gene for beta-lactamase, right? So this is your ampicillin resistance gene, the gene for beta-lactamase. The final keyword is origin, and after the origin, you have the sequence. And this sequence is basically indexed at the left end, 60 nucleotides in blocks of 10 each in per line, right? 60 nucleotides in blocks of 10 each per line is what it's given, and it is left index, which means the numbering is on the left side. So this is the sequence, but we're not going to use this sequence for our purpose. We need the FASTA format sequence. So we click here and get the FASTA format. So we select the sequence and copy paste into the box on the ORF Finder here. So this is where I'm going to paste it. And there are other methods of doing it. For example, you could have said directly taken just the accession number or the GI number or the nucleotide sequence in FASTA format. So this is the option that we exercise. They're given the nucleotide sequence in FASTA format. So the FASTA format begins with a greater than sign, followed by a small def line or annotation line. So this is basically defining what the sequence is. This is the cloning vector PBR322 complete sequence, and then followed by the sequence itself, right? If you want to annotate a subregion, you could define that, let's say 1 to 1000, but you're not going to do that because you want to annotate the entire PBR322 sequence. Then the threshold length for the minimum ORF length has to be defined. And if you look here, you have options of 30, 75, 150, 300, and 600. So 30 would be too short. We could go by default for now. We could, and then of course you can come back and change the options. So here you are. This is your 75. And then we move on and we talk of the genetic code, and we'll use the standard genetic code. Or if you want, you can use the bacterial genetic code, because we know this is a bacterial sequence. So we use the bacterial genetic code. The next option that you have is ORF start codon to use. And most commonly we use ATG as our start codon, so we use ATG only, and we're not giving any alternative initiation codons. And then of course, we are right now not ignoring the nested ORF. What is the nested ORF? And ORF within a different ORF, right? So this will be a nested ORF. So let's see what we get in the results, and then we'll modify our results to come to the final point here. So here you are, you say submit. So here, if you see, it has found 34 open reading frames. The genetic code that we use are 11, that is for bacteria, archaea, and plastids. And then, of course, the start codon we used is ATG only, right? So we're not using any other codon as a start codon. So based on these criteria, it has found 33 different operating frames, and we have not said no to nested ORFs. So you have a smaller ORFs within the larger operating frame. So example here, this is the largest ORF, or ORF5 in this case. And then, of course, you have ORF28, ORF32, ORF33, ORF23, ORF29. All these are nested within the larger ORF. And I've already told you, if you have a large ORF and other ORFs are nested within it, the large ORF is most likely to be the correct ORF. So therefore, what we can do as a first option is to ignore the nested ORFs. So then, if you see here, the ORFs are arranged in order of their size. The 5 is the largest, so it is at the top, followed by 24, followed by 28, followed by 1, followed by 20, and so on and so forth, right? And you can select the ORF here, and accordingly, the protein translation of that ORF will be shown up here. If you choose ORF28, the ORF28 protein translation will be shown here, and the region of ORF28 within the larger genomic context would be highlighted here. So this is your ORF28, if you see, right? So now what we do is we first go and customize our ignored nested ORFs, and then we also increase our threshold length to be called an ORF. So we go back here and we redefine our minimum ORF length. We keep it as, let's say, 300, and then we also ignore the nested ORFs, right? So then you press submit again, and when you press submit, now you see you'll get around 4 operating frames. So you have the graphic, and the largest one is the one that is highlighted. This is now ORF2 because there are only 4 RFs. This is called ORF2. It is a plus strand. It's on second frame. Its start is at 86, stop is at 1276. The length is 1091, and the amino acid length is 396, right? So this is likely to be a tetracycline resistance gene. So if you want to check whether this ORF is actually a tetracycline resistance gene or not, or if you want to check whether a similar protein in the database is pre-annotated and whose function you can directly take and apply to your protein of interest, that can be done here using BLAST or Smart BLAST. And here in this case, we'll use a recent reference protein or RefSec protein as our database. And first we say BLAST. So here you are, it will take you to the BLAST P page, and we'll keep everything to default. There is not much to change here. And then we say BLAST here, right? So here are our BLAST results. It has generated some ID, and then the program is BLAST P. The database is RefSec proteins, which I selected deliberately. And then we're looking at molecule type as amino acid, the query length is 396 amino acids. And then, of course, there are other things. And below this now is our match. And if you see here, the match is almost query coverage is 100%. So the entire sequence that is given is found a similar sequence elsewhere and matched to it. The E value is 0.0. The percentage identity is 100%. And then, of course, the accession length was 396 amino acids that it given. So if you look at this, this is your perfect match here, right? And probably it is to the actual sequence itself. So this now tells us that our predicted ORF actually has a function in tetracycline efflux and is basically a tetracycline resistance protein gene. Right? So now we check for the second largest ORF that has been predicted, which is your ORF4, right? And then again, we do the same thing, but we do this time a Smart BLAST, right? The protein database remains the RefSec protein database. And there you have the results in hand. So this is basically, if you see here, this gives you the highest query coverage, p-value of 0, identity is 100%, and you have maximum score of 591. And that means that this is actually the protein we're looking at. So this is, if you see, beta-lactamase or ampicillin resistance gene. So the idea then is, while ORF Finder is ab initio tool for identifying potentially protein coding regions, it also provides you the utility of BLAST and Smart BLAST to confirm those annotations by comparing your predicted ORF to known protein sequences. So we'll close here. Thank you.